Welcome to our update for the GNE myopathy patient community. I'm going to turn it over to Lolly Welsh, our CEO and Executive Director. If you have any questions for the duration of the call, we'd ask you to keep your microphones muted since we will have quite a number of people on the call today. And feel free to submit your questions in your control panel, in your GoToMeeting control panel. If anyone has any operational questions or issues, you can email or text me. I'll be monitoring that stuff during the call. And we'll send a follow-up email with some points of things that will be discussed today. So Lolly, with that, I will hand it to you.
Thank you, Nancy. Hello, everyone. I'm Lolly Welsh. As Nancy said, I'm the Executive Director and CEO here of NDF, and I've got lots of really good feedback and updates for everyone. I want to switch the order a little bit. I want to talk first about an update for the ENMC meeting that took place recently in Amsterdam, and then I'll come back and talk about the $2.5 million matching grant and what that means for gene therapy, and then Moncal and Angela can segue into the scientific piece of it. So having said that, I just want to tell you that we had the most amazing meeting in Amsterdam this year. As some of you know, we formed a consortium of all of the scientists in the world that we know of that are working on GNE myopathy. And I think at this point, we pretty much know of everyone. And last year we had our first real formal meeting at the UCLA symposium that we do annually, and we flew in the scientists and sort of combined the two meetings with giving one full day to all of the scientists getting together. At that time, Hans Lockmuller, who was then at Newcastle and has since moved to Ottawa, suggested that this year we can get the European Neuromuscular Committee to pay for it, which is basically a European organization with members who are scientists that can underwrite some of these expenses. And they co-sponsored it with NDF. And at that meeting, which was an incredible meeting, that honestly the best one I've been to in terms of GNE myopathy, we had about 30 scientists and doctors, as well as NDF and two of our CPAs, Mona Patel from UK and Maya from Israel, also attending. But the doctors ranged from everyone from Newcastle to the NIH to our own Moncal and Angela from Yale to Dr. Tassin Mozaffar, who's also on our board from UC Irvine, the Japanese, the French, the Swedes, the Israelis. And I met a new Bulgarian doctor that has 60 new patients that we had not heard of before. So I'm kind of looking forward to meeting them and introducing them to the group. I know that they are mostly Roma mutations, but it's an interesting new thing. And you'll hear more about what that means in terms of looking at the different mutations when you hear from Moncal. So that was an incredibly meaningful 3 days where we met, and while there were some differences of opinion, by the end of the meeting, everyone agreed that we would share data, that they asked us, what does NDF really want? And I made it very clear that we want the scientists to work together for the best interest of our patients. That means not waiting too long to share data, sharing it soon as they have it, that we would obviously protect their privacy and it wouldn't affect their publications, but that they really needed to build on top of one another, what one another knows. The NIH was very much on board with this. As you know, NDF has been funding the Israeli research for the past 10, 12 years. And Newcastle also came forward and wanted to ask us to sponsor and continue to sponsor the DMP natural history database, which I think is going to be very meaningful when we start to work with biotech. So it was all in all a very, very good meeting, and I'm going to send a link to Nancy or Tara that they can disseminate after this meeting, which will take you to a page that will describe who was there and what the agreements that were made. We all agreed that we're going to move forward with gene therapy, but we're going to do it in a way that makes sense. The way we've been doing it all along, again, for those of you who keep asking when is it going to start, we've been doing it. In order for gene therapy to happen, there's a lot of studies that need to take place and a lot of safety issues that need to be addressed. If you move forward too quickly, the FDA can find fault and not allow you to go forward with the next steps. So the steps are the pre-IND, which we've completed. The next step is the IND, which stands for Investigative New Drug. Then it goes to phase 1, then 2, then 3, and then it can go to market. And at each end of those steps, the FDA has to give approval for the next step. And what we realized is that if we want to be able to take it to phase 1, we're going to need about $5 million. However, we are not going to be able to take it beyond that by just being a small foundation, even with working with our friends, the Bhattacharyas over in India and all the different amazing groups that we've formed throughout all these different countries, we still are not going to be able to have that clout. And that's kind of not what we are. So what we agreed to do was that we can step up the first $5 million worth of research. If we could do that, if we could get the funding for that, then we would be able to complete the IND with a principal investigator like a Dr. Jerry Mendel or someone out of Nationwide Hospital, although to be honest with you, there's a lot of different PIs working on this kind of thing right now. And then we were going to try and find a biotech to partner with to provide us some guidance going through these early steps and making sure we use the right vectors and so on and so forth, so that when and if we get to phase 1 and the FDA is pleased with the progress and gives us the green light to go to phase 2, that biotech would then have every reason to put in their own money and to want to pick it up and take it from there. So that, as I ran it through all of the various board members and all the different scientists and all of the different industry members, everybody agreed that that was actually the only strategy that would work, and it was really quite good. So since then, what we've done is met with a couple of biotechs. Not totally comfortable sharing their names at this moment, and I will as soon as I can. But later this month, I'm going to be giving a talk to a neuromuscular disease conference in Boston in front of about 30 different biotech companies that are looking to do exactly this, working on muscle. Because as you know, muscle is a whole different game than, for example, the eye or different organs. So I really feel very, very confident that with the 2 biotechs we're speaking to right now and the new ones we're going to meet at the end of the month, that we will really be able to execute this plan. So the only remaining piece to that was, where are we going to get the $5 million? Well, due to the support of a very active supporter of ours, a gentleman by the name of David Dahl, may or may not be on this call, who's an amazing philanthropist, I was able to contact a foundation that was able to help us get this kind of funding, and they were able to give us $2.5 million towards the $5 million, only because by IRS regulation, we cannot receive all of our funding from any one source. That's
right. That's the cheapest. That's the cheapest for 2 people.
They can't hear this, right?
I can hear, yes, I can hear somebody speaking. So anyway, we basically were able to get a $2.5 million matching fund grant from this private foundation, with the understanding that we would match the balance, the other $2.5 million, and the first quarter. So if everybody could please mute your microphones, thank you. If we could, we could get the balance of the $2.5 million from everybody else, then the IRS wouldn't have a problem and the problem would be solved. So that's as good a situation we could possibly be in for any nonprofit. So we are incredibly blessed to have access to this $2.5 million fund. But of course, it presents the challenge of having to come up with $1 million by November the 30th, and the balance by February, the end of February. So what we really are hoping to do in this piece of the conversation is to let you know that the science is underway, the plan is underway. Our industrial relationships are underway. The board is on top of it, and the scientists are poised and ready to go with their work. The only thing that remains is for all of us in this community who are stakeholders in this disease. That means patients, that means family members, friends, doctors, scientists, people who make money from this disease, every single person associated with us. If we can right now put it together and ask for fundraising and ask for funding and explain that this is happening, and there'll be a press release coming out soon that you can share, then I think we can meet that goal. But I have to be very clear that if we do not reach our goal, then this will be delayed for at least another year, if not permanently. So I can't belabor enough the importance of all of us really rallying to get these funds together. And Nancy, if you could talk 2 minutes about the fundraising pages that people can do. I know they already know, but maybe you could just talk for 2 minutes about what they can do next to find out more.
Sure. So I will, following this call, send an email with lots of links for everyone who's on the call for you to follow. But some of you know that we've been very successful with things that are as easy as a Facebook fundraiser. We have a lovely couple who's donating their wedding to our cause. So instead of gifts, they're receiving donations to NDF for their wedding. You can donate your birthday. You can create your own fundraiser through our website. The trend toward fundraising now is called peer-to-peer fundraising. And that means that for each fundraiser we have, they can reach out to their community and we can get 10, 20, 30, 40 fundraisers from each person who creates a fundraising page on our site. Our board is doing it. We're asking everyone we know to do it. And it's super simple. I will send you the link. You click on Become a Fundraiser and it allows you to create your own page that you can then send the link out to everyone you know, and then we exponentially increase the number of stakeholders that we have. Our board has started it, and I think as of yesterday, we had something like 65 new donors in the past maybe 2 weeks. So it's a super effective way to fundraise, and it's super painless for you. I know that some people, it can be uncomfortable to ask, but I think that everyone agrees this is the time to reach out to companies you know, people you know, and then the people that they know. So we're going to make it super easy. I'm going to send you links to how to become a fundraiser, how to raise on Facebook, and a link to our gala registration page as well, where people may want to buy tickets to our event that's November 11th in Los Angeles or donate to the Gala Fund as well. We're going to make it really easy for all of you.
Yes, thank you so much for that, Nancy. And does anyone have any questions about the process of fundraising or about the $2.5 million grant or about what we're doing towards GNE myopathy, which of course, Monko will talk about how some of the tests that we're doing are going to tie into GNE myopathy. But besides that, besides the scientific piece, if there are no other questions, then I will turn it over to Monko. Monko, are you online?
Yes, I'm online.
Hi, it's Angela too.
Hi, Angela. Okay, so everyone, I'm going to turn the call over to Monko and Angela Leck, our very own favorite scientists from Yale. Take it away, guys.
Well, I'm trying to make him the presenter. Just one sec.
The rest of our team is also online, I believe, Heather and Karen.
That's Lolly. Hold on.
Sorry.
Turn off your camera, Lolly.
Yep.
Hold on. Sorry, guys.
Can everyone see the screen now?
There it is.
Yep. Lali, I think you have your camera on. Maybe you can. I know it's covered, but you probably can turn it off too.
Let's see.
There you go.
Yeah, turn it off. Okay, great.
New laptop, people. Thank you.
So yeah, I think everyone can see the screen. So I'm going to be presenting on behalf of the team effort that was involved in actually collecting the patient samples on the symposium day, and I think it was just a fantastic effort to get all the samples arranged. So I'll thank the team that was involved in actually getting this right at the end, everyone involved. But yeah, I have members in my lab here for right at the end, so that Angela, Karen, and also Heather, all PhDs in the lab, that they can answer any questions people may have. So this is a slide that I presented previously about the process. So one was the blood draw, and the reason behind the blood draw is to get DNA from people, so we could advance on with the genome studies and other mutation studies that we have planned, and also a skin biopsy where hopefully this is very similar to the experience that people had actually experienced, very similar to mine. So this is pictures from my experience. But while I'm on this slide, I'm going to ask, I'm pretty sure there's some patients on this call right now who actually generously donated their time and also their patient samples to this. So everyone has probably given a blood draw, but I was just wondering if someone can volunteer and tell us a little bit about your experience in terms of the skin biopsy, how long it took, if it hurt, and if there was any discomfort the next day, because I know that there are other people on this call that unfortunately there wasn't enough time or they weren't at the symposium that probably would want to give a skin biopsy in the very near future. So yeah, I'm just asking for a volunteer. Otherwise, I'm happy to jump in and talk about my experience.
Tara,
This is
you did,
I can
yeah.
I can
Oh,
talk. I had the
Yep.
skin biopsy done at the symposium, and it was probably the most seamless process ever. And I I was told that there might be a scar that would be left over, but now when I look back at the same place, there really is no scar at all. So it was just a little pinch, and they do give local anesthetic, which basically you don't even feel the pinch because of that. So, like I said, it was very seamless, and it was an easy process for me.
So, Ahmed, did you feel any discomfort afterwards, like the day after or things like that? I'm just out of curiosity for yourself.
I will say that maybe it wasn't because of the biopsy. Maybe it was just because of the armband that was put on afterwards. But I did feel a slight heaviness in my arm,
Yep.
but only for like a couple days, and then even that wore off.
That's a curious point that you brought up. So when I gave a skin biopsy, it was from you can see from my lower leg. So yours was on your arm. Whereabouts on your arm, out of curiosity? So others that haven't given know.
It was right on my shoulder,
Okay.
like
Great.
yeah, by my shoulder muscle.
Yeah. And if you're going to give any additional feedback on, if anything could have been better in terms of just making it more convenient for you, would there be any feedback on that, out of curiosity?
I wish I could find any other way to make it better, but I mean, it was just so easy that I can't think of anything that could make it any better than it already is.
Moncol,
That's
may I ask a question real quickly? Are you
Yep.
looking to get more samples from more people? Because do we have to send people to the UCLA facility to do more samples, or are we looking to actively get more samples right now? How are you thinking
Yeah,
of these?
I think it'd be fantastic to get more samples, and we know that the UCLA facility did a fantastic job in terms of, as Ahmed said, the process itself, and also getting it safe and sound to us here at Yale, so we can process it further, which I'm going to touch upon in a little moment. But yeah, so the UCLA facility, and there's probably Lali, you would know better, the collaborating physicians in California, like in Irvine and things like that. But, I think the symposium was a great event to have because all the patients and their family members are there. But, I think it doesn't have to be just at the symposium. My thoughts are.
Yeah, it would be really great if, for those who missed out on donating sample, the next time they are in the UC Irvine area
Or UCLA.
or UCLA, yeah. I know Tassin and also Dr. Perry Shea have offered to collect more. I mean, the more samples we have, the more power to our study, and the more we can be certain of the conclusion and the results that we get from our study. So definitely,
Yeah.
yeah, we would love for more if possible.
Hi guys, this is Lale again. I just want to jump in real quickly. I was talking to Tahsin Mozafar, and I understand that Perry Shea may have some sort of an agreement that covers this as opposed to UCI. So at this
Okay.
time, we're just directing people to Dr. Perry Shea, but we
Okay.
will move forward in terms of collecting other samples. How about for patients that are on the other side of the world or the other side of the country? How can we, is it possible, and this is perhaps a discussion we can have another time, but is it possible for them to donate samples in a way that's unified in the same, collection process and all of that stuff, Monko, Angela?
It will never be fully unified, but Ahmed also would like to sample from the same place. Like if it's the shoulder, then we'd probably want to do that. But the procedure should produce very similar samples anyway, because it's not such a complicated process in terms of getting skin biopsy. So yes, we can do it. I think the only challenges is like what the UC Irvine site has, is if their physician or the clinic they go to have research protocols that allow them to take this from the patients. But other than that, I think that's the main barrier. It's the research protocol, and to have a nurse or a medical professional that can actually perform the biopsy also, which there normally is at many clinics. So it's more the research protocol in place. But if there are patients here that are not from the West Coast, definitely just drop myself or Angela or someone an email, and we can kind of coordinate that, because I think, as Angela said, the more patient samples, I think the more power we have to do research on it. And it's not just one research group. It's the thought to make this available to all research groups that work closely with the NDF, and also new research groups that want to work on GNE, because we're creating this resource to advance GNE research and not to benefit any one researcher, and so to be a lot more patient centric is sort of like the thought behind this.
But if anyone is thinking about donating samples and they are overseas or even in the country, please let us know beforehand so that we can coordinate with your doctor or whichever hospital you will be attending, as we will need to prep them on how to collect the samples. And yeah.
Cool.
So I've just put both Dr. Leck's email addresses up on the chats feature for you, so those of you who don't have it can take it down. And thank you for answering those questions for us.
Yeah, no worries, Lolly. So this is just to remind everyone, we collected skin biopsy, blood samples, and affected patients and their first-degree family members also. I think the next day after collection of both the biopsies and also the blood samples, they were safely received on the Yale end by Dr. Heather Bess and also Dr. Karen Woodman, and you can see a picture of how the samples look like. But this is the blood samples in the freezer later on, so they were put on ice pretty much immediately while the biopsies were processed. And one of the things we did with the biopsies, well, half the biopsies was to produce patient cells. So these are de-identified as per the research protocol. So unfortunately, people on the call can't say, hey, these are my cells growing. And this is just to protect patient privacy. So everything's de-identified, where only people who are part of the research protocol know who the samples belong to, which we don't. Most researchers only need to know what the mutation is, the age of the sample, and the sex of the sample, and not necessarily the patient it belongs to actually do research on it. So here's two different patient samples at different magnifications, and these are fibroblast cells or cells derived from the skin biopsy.
Yes.
So Heather has left me notes to say that I think these are pictures. I think these are pictures from more than 3 days, but Heather can correct me if I'm wrong. But but some pictures were taken a few days after they were grown, and I think these are more recent pictures where you can see there are a lot more cells, and they for me they look quite healthy and growing well.
So basically, what this is that so the skin was taken out from the shoulder, as Ahmed mentioned, and half of it was processed in our lab, where Heather and Karen chopped them up really finely, minced them into little pieces, and sort of used an enzyme to break the skin apart, and then try to extract cells, individual cells, which we can then continue growing in the lab. As you could see, these are a population of individual skin cells. And this is something that we can keep growing in the lab under the right nutrients. We can sort of trick them to believe that they're still inside the human body. And from that, this is kind of a snapshot of what's happening in a GNE patient's a body. And so we can then test therapeutic compounds, we can test different things to see how the cells respond and behave. So, Monko, do you
Yep.
want to?
Yep, so I'll go to the next slide. Is there any questions regarding the
And-
patient cells?
And these are cells that can be then frozen down to sort of freeze them in time. And they can also be sent to any scientific collaborator who has any interesting studies planned or any potential therapy that they want to test on any of these patient cells. So this is an NDF sponsored and NDF shared resource that we will make available to the scientific community.
Could I just ask you
Yep.
guys to refresh everyone how this will help with the end goal of gene therapy, like how to tie this into gene
So Lali, I'm going to touch upon that after presenting the samples. So I'll get to that to remind everyone, who've generously donated.
I see. Okay. Well, then there's only one other question on here, and that's somebody, Eitan, who says, is it possible to interest Bill Gates in this study? I suppose anything is possible. If you guys have an in with Bill Gates, by all means.
Yeah. But
Yeah.
but on that point, I think to get others interested in the study and to donate, funds so that the research can advance. One of the things, beyond natural history studies is to have a good set of patient samples or a what they call in research a biobank, because then a lot more can be done. And creating these things is hard work and takes a lot of time, and but you have to start somewhere. So Karen and Heather have created 13 cell lines from the 13 from the skin biopsies, and I think it's a great start. And I think over I think the next few years that will rapidly expand, especially if everyone has positive experiences with their skin biopsies. So what's sitting in the freezer and waiting to be processed at the moment are the other half of the skin biopsy, and that's for RNA extraction. So RNA, as we've presented in 2 previous meetings, is another way of looking at the changes in patient samples compared to, say, their healthy siblings or healthy people. And it's a way of developing biomarkers, but I'm going to touch upon that in a future slide. And what's also sitting on ice is the blood samples, where we're only using it solely for DNA extraction in this case. For future studies, we may have, if we had more time, we could plan for the blood and also the skin to be preserved in a way that would be appropriate for other studies too. But for this time, this collection round was a pilot run to see how the logistics work, and it worked actually a lot better than I expected. It went very smoothly. But to perform RNA extraction, DNA extraction, it should be a 1 to 2 day process. So it's not a lengthy process. And although they're on ice, Heather's left me notes that what's critical for the quality, maintaining the quality, is how quickly we put them on ice after we receive them. And so these were put on ice very quickly. So the samples are fine at the moment in -80 Celsius freezer. And then the next thing we want to do is create the DNA and RNA, which then it can also sit for a very long time without degrading and be available to researchers. So it can help them better understand the genetics of GNE myopathy. Cool. So in terms of the next steps, one of the next steps is, and this will be a quick process, so what we want to do is obtain extra details on the patient donor. So a lot of that Perry would have, and his staff in the UCLA clinic would have already collected. And there are 4 critical things we want to collect, whether it was from an affected patient or their family member, what mutation that patient had, the sex of the patient, and also the age of the patient at collection. So none of these details, I believe, are what we call patient specific identifiers. So we maintain privacy based on that. And because we're not collecting date of birth, people's addresses and things like that. And this is probably the bare minimum researchers need to know to work with these samples and actually make sort of like meaningful discoveries. And also by collecting these 4 pieces of information, it allows us to validate everything, that sample with the de-identifier belongs to the patient we believe it belongs to, and we can validate this and infer this from the patient sample. And it's a way of just making sure that we're producing a high quality biobank, because I think anyone can produce a biobank and collect patient samples, but it's the quality that you maintain that makes others interested in actually using it for research. So the next thing is RNA extraction from the biopsies. And just a reminder, the reason we're doing this is actually to see any differences between patients and healthy samples. So what genes are they expressing, and at what levels that may be different between patients and their healthy samples. And that allows us to look at what we call biomarkers. And biomarkers are very important when you're developing therapeutics such as gene therapy, as a way of measuring if any therapy such as gene-based therapies are actually making a difference in treatment for the patients, because these things will move first, and we will have more sensitive measures to detect those movements before we see any benefits in muscle and day-to-day life. The next is the extraction of DNA from blood. So a lot of these samples will probably potentially be used for whole genome sequencing project, because this DNA comes now with patient cells, it comes with patient RNA, and it also comes from a patient that we know is a very good participant and good donor, that we believe that if we make any findings in the whole genome sequencing, we have a lot of data and patients' samples to actually follow up on that finding. And the last thing is the cell lines from skin biopsies. So they've already been created in the lab. So next thing is the validation of the GNE mutation. So matching up what had been collected from the patient with what we actually see in their cells. Again, this is just to ensure quality and consistency of the samples. There's a possibility then, once we have the fibroblast, to create muscle-like cells and other cell types. And this is a way of just seeing if, say, we discover biomarkers, whether they are applicable also to muscle-like cells, also that they are different in those cells too. And it also allows us to test gene editing approaches. And one of the things that I had presented previously is CRISPR-based editing. With CRISPR-based editing, at the very least, what we can create is patient cells with the mutation corrected, and therefore after correction, that they make for what we call in science a very good control. So they make for what we believe should be a more healthier cell, and we can have a look at what effects that has on the RNA expression and things like that. But none of these things would be possible without the patient cells. And it was something that I think it wasn't that the field didn't have, but it was just much harder to share. And so I think there's no use in collecting this if we don't create a framework and also a mechanism to actually share this with other researchers in a way that's a lot easier for them. And so, so I think another future step would be to work with Lali, Nancy, and others in the NDF, including the Science Advisory Board, to to get their advice on how to make this best available to everyone that wants to advance GNE research. So before I transition to the next slide, to remind everyone what we can use these samples for, using another muscle disease as an example, do we have any questions regarding next steps?
I think we
Hi,
did.
my name
Oh,
is Mangesh, and
Yep.
I did have a question actually about the previous slide. I think I must have missed some context. It
Yep.
is not clear to me why the fibroblast, or maybe even another slide before that, sorry,
Yep.
why the fibroblast cells are so interesting to GNE myopathy research, or in general to muscular dystrophy research.
I think that's a great question. I'm going to attempt to answer it, but I'll get others in my lab to jump in who work with fibroblast cells more. But I'm going to respond to this in terms of a practical point of view. So yes, muscle biopsies also would be great to get, but there's always a balance between inconveniencing the patient and the pain it causes. As Emad had said, that the skin biopsy was relatively quick and painless. Well, relatively, emphasis on relatively. But I know from myself, I've given a muscle biopsy, being a muscle disease patient myself, and I can tell you a muscle biopsy is not quick or painless. It's actually very painful. So by collecting fibroblasts, we can study aspects and quickly create muscle cells from it too. So it's a balance between pain and sort of like on the patients. The other thing fibroblast allows you to do is fibroblast is one of the cells of choice of creating induced potent stem cells. So fibroblasts are the cell type of choice, and when you create these special stem cells, you can actually create other types of cells from it. So it's a balance. So you're right. And the last thing is by creating, we know from projects that I've been involved in my previous life in Boston, that the GNE gene, yes, causes a muscle disease, but it is also highly expressed in the skin. So it's another way of getting at something, at a tissue type that's less inconvenient to get to and also expresses the gene of interest.
Yeah, for some reason, yeah, we were actually really surprised to find out when we looked at some databases that the GNE gene is actually more highly expressed. So it's more, it's switched on more in the skin cells than actually the muscle cells themselves. So that's, you know, for if we want to test gene editing strategies where we fix the patient mutations and then measure the output, such as levels of GNE expression levels, I think fibroblasts would be more informative than a muscle line. But yeah, as Malcolm said, I guess the most powerful point is that fibroblasts have the ability to be converted into muscle cells by triggering the right factors. And scientists have figured out how to do that. So even though initially we have fibroblasts, and skin cells, we have the means to turn them into muscle cells.
But yeah, I think that's a great question and a reminder to put some slides to do a compare and contrast of extracting a skin biopsy versus a muscle biopsy, and also, you know, the strengths and weaknesses of both approaches. But does that sort of answer your question?
It does. Thank you. I think that's extremely useful. A couple of follow-ups, if I may. One is that, is there any research around this that's in the process of being published or already published? I think references to that will be great, because I think just in in terms of research papers, trying to figure out like how these cells are used
my dear, so you want the account to take you to 1 hour, 2 hours?
I'm
Dave,
sorry, could someone
Can
please
someone
put
mute?
your phone on mute?
So the question was if we can share some publications. I think yeah. So I can ask Nancy to get us connected, or I think you have my contact details, so you can just email, and I can show publications where they've looked at RNA expression using microarray, which was the technology before RNA sequencing, and also previous studies on fibroblasts. But again, this is an effort to create and broaden out that resource for the research community. Was there any other points? You said you had
No, I
a
think
few.
that's great. I'm not doubting it. I was just looking
Oh
for
yeah. So
like
yeah,
what
yeah.
the other follow-up was that for some of us who have, I think, done muscle biopsies in the past as part of our diagnosis step, I wonder if any of that data could be shared with your team. Would that be helpful? Again, maybe that's like a slightly separate topic, but I would love to follow up with you and Lale and Nancy.
Yeah, I think it. Previous muscle biopsies are good for RNA sequencing, but I think we need fresh muscle for to create cells from it if we were going to create fibroblasts and muscle cells from it. But what we can take from preserved, previously extracted muscle is to do RNA sequencing. So it may be helpful if we want to know that some of the biomarkers or the changes we observe in skin biopsies are also relevant in muscle biopsies. So they may be relevant in muscle cells, but ultimately it'd also be good to know if they were relevant in muscle tissue, so which means the muscle biopsy itself.
Yeah.
Thank you.
Cool. So yeah, hopefully answering those questions are useful, and it is a great reminder to actually put those slides for future talks. So thank you for the feedback. So I'm going to transition to this, and I'll let Angela talk about it because it's her slides. Just a reminder of what we can do if we have patient cells, and what we can do with biomarkers.
So I studied this other form of muscle wasting disease known as FSHD. It's a type of muscular dystrophy that also affects mainly adults. So it's a late onset form of muscular dystrophy. And basically, what we did in our research consortium with a few labs, partnering up with a medical doctor who took biopsies from her patients and also the unaffected relatives, is that we were able to get a set of genes, these here that I've highlighted, are genes that are strongly turned on in the context of FSHD in patients. So some of these genes, which I've highlighted here, are known to be common FSHD biomarkers that now everyone in the field likes to use in their testing when they test, for example, different therapies. So these are 3 of the genes here listed below, the FSHD biomarker, as I've mentioned. In patient cells, these are elevated. And then when we test different therapies, the effective ones manage to lower their levels to baseline, to what we see in healthy control cells. So this is an example of a way we can use biomarkers through, for example, RNA sequencing of the patient cells in order to inform the scientists whether a therapy is efficacious or not. So
yep. So with that, I just want to before we take questions, and because we have Kieran and Heather on, I definitely want them to answer the remainder questions, especially if it's specific to the sample processing or next steps also. So, but I'd like to say a massive thank you firstly to all the patients and family members that volunteered. So we would not have patient samples if no one volunteered on the day. And it's amazing to get so many volunteers on the day, much more than I had ever hoped and guesstimated. And also just to let the patients and their families on this call know one thing, that Angela came up with the idea of actually building up this resource, and the symposium was coming up. And normally with a lot of research projects I'm involved in, and also patient symposium, this never ever can be done in such a short time. I think it was only 3 to 4 weeks. Normally, this takes about a year in the research environment, because people love to throw paperwork at people rather than think about how can we make this work for the patients. And I'm not kidding. I went to a symposium a month after this one, and I suggested the same thing, and everyone just laughed at me, going, this takes a year to prepare. So, and just to emphasize again, what takes a year to prepare is all the paperwork. So it's a massive thank you to the guys in, first of all, in UCLA, particularly Dr. Perry Shea, who didn't think that, you know, these things should take a year to prepare in terms of the paperwork, and also the research coordinator in his group, and also members of the clinical staff in UCLA that actually made this happen and didn't take a year to make it happen. Also the NDF. So one of our biggest concerns was where the clinical center was compared to where the conference was. And Lali and Nancy and others at the NDF were amazing, going, we can make this work, we can make the logistics work also. And, you know, and as I can see that it actually worked. And a big thank you for people in my team, Karen and Heather, who you may not know, but it is a lot of work to actually process that many samples and get it in a state where it can actually be in the freezer, or to expand out the cells for 13 samples. It can take weeks to a month, depending on the condition of the cells. So depending on the condition, the biopsy, the age of the patients, and a few other variables. So there are variables in actually processing it. So I think the longest one took over a month to actually prepare. So they had to be looked after like a pet. It had to be fed. It had to
like a plant.
Yeah, like a plant, not a pet. So it's a lot of hard work. And also Angela,
or Anglia.
Anglia. Yeah, Anglia and Angela. For the coordination over everything. I did very little, but present these slides on the behalf of everyone that was involved. So it's a massive team effort, and just let the patients know that but they have a great team, you know, who've made this work in such little time. And I'm very excited to see, to work as a team with the patients to actually build out this resource so we can advance GNE myopathy research. And I think, you know, if I haven't emphasized enough, like Lali, that having these patient samples will produce biomarkers and things that will be highly critical in terms of assessing whether gene therapy is working and how to actually make gene therapy better in its development.
Yeah, and it's something that the FDA will very much appreciate and look very favorably upon when the gene therapy is being proposed in the IND. Because they'll ask how you're going to measure if it's going to work. GNE myopathy is associated with a slowly progressing muscular dystrophy. You don't expect the therapy to result in, you know, patients walking straight away. How are you going to measure if it works? So, you know, one way is by measuring these biomarkers. So yeah, thank you so much, Lali and Nancy, who are always, you know, how can we do this? How can we get it done? How can we, you know, even though there were so many logistical barriers and just things like that made this a really difficult endeavor. I mean, they powered through, and they were so resourceful, and you guys are very fortunate to have them in the NDF and leading the way for you all.
Cool. And with that,
sorry, we have Yeah, sorry. I was just going to say, have you seen the questions? Can you see the questions that we have? I'm sorry, maybe you're not ready for them
No,
yet.
hang on. It's probably in this chat
Okay,
thing.
I'll read them to you. We have
See the questions if you want, Angela and Monkol. So I'm Karen, I'm from the LEC Lab as well. So the questions we've been asked is, can we get a copy of the slide deck? So I'm sure Monkol, we can send that out.
Yep.
With my name correct, please.
Yeah.
With your name correctly spelled on it, Angela. The next one is, when will the RNA-seq data be available?
I'll jump in and answer that. So I think the first thing will be just to get approval from the NDF to convert RNA, and then probably either our research group or another research group will put in a research proposal that will be approved by the NDF board before producing that RNA data. But it's the hope that a lot of the things that we produce will be shared amongst all the researchers. The data itself wouldn't be as useful to the patients in its rawest form, but the results from the data, the RNA sequencing data and the analysis would be more important. And we hope that whether our group or another group actually performs the RNA sequencing in the data analysis, that they would actually present it to the patients in a forum such as this.
I think that the RNA sequencing will benefit from more samples, and I think that I would want to wait to submit the samples for RNA sequencing when we have more samples, so that they can all be processed together under the same conditions. Because with RNA sequencing, it's really difficult to combine different batches of patient samples that have been submitted differently under different conditions.
Exactly.
So, yeah. So I want to do the RNA sequencing all together when we have enough patient samples to get a meaningful outcome. But I think that what we will attempt first before the RNA sequencing is the gene editing approaches on the individual, where we'll pick patient cells that look like they're amenable to gene editing and fixing of their GNE mutation. And that's something that we can attempt firstly before the RNA sequencing.
So I'll jump in and add to that. So we have designed on some of the more common mutations in GNE, which ones can actually be edited. So we'll be trying that first, those mutations first.
Can I also jump in on behalf of...
Oh, sorry, Lally, you go ahead.
Oh, sorry. I just wanted to say really quickly, based on what Monkol and Angela were saying, that from our process, it's important for our patients, and I believe it was Alok's question, Dr. Bhattacharya, I want to let you know what our process is internally. Perhaps it's interesting to all of you. When a proposal comes in from us, we have only a 6-week window for our scientific advisory committee to review it, and then they give back anonymized feedback, which we then compile, give to the board, walk the board through what that means and what the implications are. The board then votes on it. This is all compliant with IRS regulation. And then we quickly get back to the applicant and fund it ASAP if it's a go. So absolutely without delay. I know that with a lot of nonprofits, things can be quite slow. You will not expect slowness on our part. We are extraordinarily impatient for that sort of thing to happen. So, but it does have to make sense. And what Angela said about waiting till she could compile it all makes complete sense to me. Sometimes what appears to be the slowest approach may in fact get us to the best results fastest. And then I also wanted to ask if you guys could talk, Monkol, Angela, and your staff, maybe about how this will tie in with the whole genomic sequencing project that's already underway, and how will that, build on that for you? And then I'm going to mute my microphone.
Yeah, so I can answer that question, Lally. So how it will feed in is, so just to reiterate, some of the things I touched upon during the presentation is, one, some of these samples will be used for the genome sequencing, because now that we have a fresh blood draw and therefore create fresh DNA, this will probably be the nicest, highest quality DNA produced. So some of them will be, if they fit the inclusion criteria, which I believe some of them will, they will be included in the whole genome sequencing. And what that will allow is that we have this perfect match of genome sequencing data with the patient data where we have patient cells and also patient RNA that we can follow up on any interesting result. And also what that means is even if we don't have that perfect match, we do have patient samples that we can follow up on interesting results. So these patient samples that match may have the same mutation also as the interesting results we discover. And what we're trying to discover is mainly what are the genetic causes of some of the discordance that we see in patients, where some are more severely affected than others, or some have the disease a lot earlier than other family members. So that's how it ties in. And I'm going to answer the next question because it's kind of related to what Angela touched upon in terms of when RNA sequencing data is going to be produced. And it's Mangesh's question about how urgent and useful is it to get more patient skin biopsy specimens. You know, I won't answer on the urgent, but I think if we can get the numbers up a lot quicker, then we can actually sort of like queue them up for RNA sequencing, mainly because yes, we can do it in sort of like a tiered fashion where we're just sequencing them as they come. But I know from my experience of working with RNA sequencing data on some large projects that I worked on at the Broad Institute, that you will get a lot of batch effects if you do it that way. So it would be good to get a good number for the first, say, pilot run and produce a lot of good data for the research community to use.
Dr. Monkol, just a quick follow-up question on that then. Does it sound like UCLA is the only facility where we could donate or we could get skin
Yeah, that's
biopsy
a
done?
great question. That's a great question. So I think someone asked this earlier also. So it's not the only place, but UCLA is the place where we know that they have the right research protocol. So so it's a combination of two things, whether they have the facility to do the skin biopsy, and I think a lot of clinics do have that. It's actually the other thing that actually is the main barrier, whether they have a proper research protocol for them to take patient samples and consent the patients to that research protocol, which Dr. Shea did have. They had a general biobanking research protocol that they had used for this project. So yeah, the research protocol is generally the main barrier, and that's why I was saying that normally planning these things take about a year. I can go on about my complaints about research protocols and why a lot of times it's not in place to actually help patients, which is ironic, but you probably don't have 2 hours of your afternoon to hear me complain.
And I hear what you guys are saying, and we are going to, just to tack on to what Monkol is saying, we are going to try and figure out this relationship with other key locations. But as Monkol points out, there are a lot of legal and practical issues related to that may not make it as easy. But we will continue to try, and perhaps with the help of Drs. Bhattacharya, we might be able to get an Indian cohort of samples, somewhere where there's a lot of patients, but also maybe another site in the US. For now, we will work on that, but just so you know, it's harder than it sounds to set that up. So thank you for your patience, and we will continue to work on it.
So one of the questions is, do you have a tentative timeline for conducting gene editing research on fibroblast cells or related muscle cells? This has been the biggest unknown in terms of whether in using the cells for that research purpose we can share. So that's a great question about the timeline. At the moment, we haven't set any like strict timelines on that. And Karen has corrected that we're not correcting stem cells, just it's just the fibroblast cells, which could potentially be created into a form of stem cells. So the fibroblast itself will be corrected. But we're hoping to push that. I'm not sure if there's any more questions.
Yeah, so I just responded to Tara's question, which she asked whether GNE gene is highly expressed in skin cells. Would it be applicable for stem cell therapy? So that was what I responded to.
Yep, great. Thanks, Karen.
I don't have any further questions, but just wanted to say thank you. This was a very useful presentation. I think it's great to hear all this research that's going on. Thanks again.
You're
Cool.
very welcome.
Yeah. Thank you very much for some of the great questions and their great suggestions on how to improve the presentation, what to include for next time. So thank you very much.